Hi, my name is Venkatraman Siddharthan. I'm here working at the Institute for Antibody Research in Utah State University. A small brief about me. I'm from Trichy. I did my bachelor's and master's from Trichy itself. And I moved to Chennai to do my MPhil in endocrinology. And then I went to All India Institute of Medical Sciences at New Delhi, where I did my PhD in the Department of Anatomy and specialized in the neuroscience. And I moved to after that, I moved to the United States here. And before moving to here, Utah, I worked with the Johns Hopkins for 2 years. And most of the presentations will cover whatever the work I have done so far. I think you will know more about me using the presentations, what I have worked. On your right side on the screen, you will see this modeling the human diseases at a small abstract, which I provided. What I'm going to talk on this presentation, it's a synopsis of my presentation. If you see that, I would be talking about mostly on virus, because I've been working with the virus for the last 15 years. I'll be taking as an example for this, how we can use these models. So that's the purpose of this using the viruses here. So we'll talk about the infectious virus manifestations and that resembles disease. And we also will talk about the therapeutic approaches using antibody and antibody treatments. And possibly I will not be talking about the active dependent activation, the last one, which is bottom of the my abstract. So now let's move on to the presentation. Okay, here, as we all always start about this history of where we started, how we started, So the same way I would like to start, on your left side of the screen, you will see the researchers, they are from the French scholars. They valued physiological experimentation in service of scientific progress. So, and you see that the people, those who are looking at the dog, the heart open dog, you don't see any expression or anything. They're all curious to see what is there. But when you see on the other side, on the right side of the screen, there is an experiment. Again, this experiment is being done. A bird is kept on the pump, a live bird. People, those who are surrounding that, they're all thinking about what's happening here, why we have to do that. There is anxiety, sadness, some are looking sadness, very sad. Some are looking, talking about the non-responsive. So we have both research and activists going on all the time. So which we are having right now also. So these pictures are still welcome. So whatever the case, the human and animals would still be dying from rabies if Louis Pasteur had not experimented with dogs. So the animal is kind of always necessary to move on with the treatment. Okay, biological models. Basically, biological models are classified in two different categories. One is in vitro and So in vitro, we can have two-dimensional cell culture model. Just I'm trying to mute people here. Okay, so in my presentation, I'll be talking the two-dimensional and three-dimensional in vitro cell culture model. And two-dimensional is the kind of you see the breadth and width of a cell. That's two-dimensional, and the later one was the three-dimensional, which has been physiologically relevant things have been used more than one cell type to do the cell culture. So that's why it's called three-dimensional. Others, the previous one was the two-dimensional, which is the golden technology where has been done for many years. And organoids and spheroids are the different type of cell culture. Organoids are the cells. Stem cells are being used to culture that. Spheroids are the tumor cells has been used to culture. I'm not going to involve on that. So let's move to in vivo. So the bigger one, where we have been using the invertebrate chordates. So in the invertebrates, most of the master students and the research scholars would be knowing that, heard about this Drosophila melanogaster had been used for many years for different types of the genetics. And the other one is the nematode, C. elegans. There are many other things that have been used in invertebrates. I'm just pointing one or two here. C. elegans, have a very good similarity. You will be surprised to see that. Maybe by the end of the presentation, someone will Google it and find out what the relationship between the C. elegans and the humans. You'll be surprised to see that they are 1 millimeter in thickness, and that has been used for many of animal model studies. And in the chordates, all the phylum animals, fish, frog, and everything had been used as animal models. We'll talk about that one by one as we move on to the presentations. And the latest one is in silico, is using computers. People are. Studies have been done using in silico. But the only thing is that, you don't have any ethical considerations as long as until you publish the manuscript of using in silico model. Okay, now I'm going to introduce a series of new things like this one, but I'm thinking this is not a new one because, because of the pandemic, we all know that what's a virus and how a virus is getting into the cells and how we have to treat, what kind of vaccine has to take. We have been educated by this media for the last 2 years about this, virus and stuff like that. But still, because of, for the presentation purpose, I have to introduce a little bit of this life cycle of a virus. So as you see here, these are the small virus, and this is a body cell or host cell. The first step would be the attachment and entry. If the virus enters a cell, and then subsequently the cell begin to strip off the virus outer coat of protein. And then the nucleic acid in the center of the virus is being released. And the next step is the viral genome replication and transcriptions, where the nucleic acid gets to the cells of the host and start chemical manufacturing system. The next one is the translation and protein function, where the cell ignores its own chemical needs and switches to making new viruses. And finally, viruses are released at the expense of killing the host cells. So this is a normal life cycle of the virus, but it will be very little bit here and there, but otherwise it's the same. This I got it from this. Okay, now I'm going to introduce another one, curcumin. As we all know that, we have been using this one, turmeric, for the last since 250 BC. I heard that in the Ayurvedic, that Shushruta Ayurvedic, that has been used to relieve the effects of poisoned foods in 250 BC. You never know that, what it would have had in those days, and people have used it to do some lot of work. And we all have been using every day, day in and day out, in the food and all sorts of things. I know that, in southern part of India, we have been using a lot of this turmeric for everything. So the main ingredients of the turmeric is the curcumin, and in this study, here I'm using this as an example how turmeric can be used here to block the SARS-CoV-2 infection on the cell. So I'm taking as an example for the two-dimensional cell culture. So the curcumin has three activities. One, it's prevent the viral binding to cell. And then it also involves the immunomodulatory function, like it can modifying these. It's modifying the IL-6 and IL-1 and TNF alpha release. Those are the cytokines, and that's by preventing the oxidative stress and leading to the preventing this apoptosis of the cell death, and following preventing the pneumonia and ARDS. And then now I would like to okay, I'm sorry, I skipped one slide. Okay, I'd like to thank Mr. Thiru S. Shanmugam, who is the director of the Polyhedron Laboratories, and who is the one kind of initiating for the Green Pharmaceutical Initiative, and he is our very, very good family friend. And I don't want to go in detail of that. And he is an excellent friend of my family, and who is the one who, whenever he talks to me, he calls me, he would like to have something like a to be initiated, like a Green Pharmaceutical Initiative, making sure that people are using these, increasing usage of phytomolecules and their analogs as drugs. So as it was also been said by these, on this Dr. Rajendra Prasad readings, opening notes, and then it was read by Professor Bonvi. So I really thankful to Sivashanmugam, and I'm really thankful to all the participants. Without you guys, and very thanks to all the colleges and faculties, research scholars, undergraduates, and master students. Thank you for all joining on this big initiative, Green Pharmaceutical Initiative. Okay. Now let's move on to the presentation here. So for the two-dimensional cell culture here, what we see here is the cell culture flask where the cells are being grown. And then here you see, once the cells being confluent, that will be moved on to the 96 well plate. Okay, so this is part of this experiments and how it has been used. So once the cells are being confluent here, and then they seed the cells here. Okay, the cell like a well like this. So it will be grown for 24 hours, and then the virus will be introduced onto the cell for 1 hour, and then they remove the virus and they add this turmeric, concentration of turmeric for 48 hours, and then they harvest supernatant. Once they harvest supernatant, they are in the plaque acid. I will talk about it in detail because right now it looks like new thing, but let's play the video and see how it goes.
Inflammation, low-grade inflammation, is the underlying factor in over 90 to 95.
Although viruses are typically too small to see, if infection causes visible damage to cells, the presence of infectious virus can be detected using a plaque assay. We're going to be looking at a plaque assay here carried out using influenza A virus. We need to have indicator cells relative to infection. Flasks in 6 well dishes. By the next day, a confluent lawn of cells, a monolayer, will have grown. Take sample containing virus and carry out a 10-fold serial dilution. These times will span a range of dilutions from 1 in 10 right down to 1 in a million. Then washed and infected with the whilst the infection is taking place, the phosphate buffered saline dissolve it. This is done to increase the viscosity of the overlay suspension. Normally, newly released virions will diffuse through the media and infect all of the cells in the dish. But by increasing the viscosity, by mixing media with agarose, we can ensure that each infected cell only infects its immediate neighbours. Once the cells in the gel are returned to the incubator, the infection proceeds. Influenza grows relatively quickly, and areas of infection will start to spread over a course of 2 to 3 days. During this time, each cell infects its neighbours and they infect their neighbours, and you can see here the growing zone of cytopathic effect. This is, however, quite hard to see with the naked eye. To make it more visible, the overlay is peeled off and the cells are fixed and stained. Here we're using Kumasi brilliant blue dissolved in ethanol and acetic acid. Once this is washed off, uninfected cells are stained strongly blue in a confluent layer. However, any initial site of infection will have left a visible plaque, an area where no cells remain on the dish, which appears white against that blue background. In an infected plate, we'll see now a range of different densities of plaques across the serial dilution. And by taking a well with countable plaques, the number of plaque-forming units, or PFU, in the original stock can be back calculated and expressed as a concentration of PFU per mil.
So that's how it has been done. You've seen that they've started culturing the cells on a bigger flask and they seeded the cells on the 6-well plates. So that's a cell culture, 2-dimensional cell culture. And then they, at the end of the washing, they showed that there's a blue color On that, and then these are the whites or the dead cells. Wherever you have the dead cells, that will form a hole or a plaque like this. That's called plaque. So this is untreated control where there is no curcumin added. So you see that there are a lot of cell death. Whereas in this is a kind of positive control, hydroxychloroquine or chloroquine has been used here as a positive control. It has some mechanism of actions early on the infection. So that's why they use this one, and it's a positive control. Coming to our compound curcumin, when they add 10 micrograms, you see the less of the plaques. And as you move on to the lower concentration, you see the more. So this author have already done the 2 or 3 different experiments where they've done here, you see the post-infections, but they also done the pre-infections and co-infection, meaning the co-incubation with the virus and curcumin, and curcumin has been introduced before the virus in alcohol. So that's called the before the pre-infection, co-infection, and post-infections. So based on all 3 steps, they found out that this affects the SARS-CoV-2 with the replicative cycle of the virus. So this is basically an example of a 2-dimensional cell culture where you grow the cells, do the virus infections, and you also do the plaque assays. And above all, the curcumin, we are using it, as I said earlier. So it has a lot of antiviral properties against many of these viruses here. But mainly just remind everyone that this has been done in the cell culture only. Some of the studies has been done in vivo, some animal model, but mostly the data what we see here on the screen is all from the cell culture model. Okay, now I'm going to talk about the 3-dimensional cell culture model, where I'm going to introduce another new terminology called the blood-brain barrier. What you see here is the cross-section of the brain capillaries, where you see the blood in the capillaries, inside the capillaries with the leukocytes. And you also see the pink cells surrounding the blood. That's the barrier. That's a cellular barrier which prevents the entry of any molecules crossing the or going into the brain. As we all know that brain is an immunologically privileged site, meaning that none of these cytokines or pro-inflammatory cytokines can be enter, or any virus, any bacteria can cross that. So that's the barriers. So that's made by these cells. Okay, this cell otherwise called endothelial cell. Okay, so surrounding that endothelial cells, you see that green color cell, that's a pericyte. But on the outside of the basement membrane, there's a cell called the astrocyte, which is the supporting cells for the, which we call as supporting cells, associated microglial cells. So this also giving the, touching the basement membrane. So what is the purpose of that? So the main idea of this 3-dimensional model, this I have developed at the Johns Hopkins, to understand what is the role of the astrocyte, what kind of factors is being secreted from the astrocyte, which helps maintaining the barrier properties. Okay, for that, I'm going to introduce another thing called transwell inserts. The whole thing was done in the transwell inserts. What is the transwells? So what you see here, this is called the transwell. These are the semi-permeable membrane in which you can grow the cells. Okay, so basically we have, as I said, just to mimic this one, you have the endothelial cell here. So the same thing, endothelial cells here, and you have the blood inside. So I introduced the medium to flow this one. So that will create a kind of shear stress, the flow stress, that's called shear stress. Cells will be exposed on that. On the bottom, we have the astrocyte like here, just to mimic that. So the way we did that, first we invert these transwell inserts and add the astrocytes to grow for a day or 2. And then once we see the semi-conformant, we flip the transwell inserts, and then we add the endothelial cells on the other side and let it grow for 1 or 2 days. And then we introduce them everything onto this chamber. Okay, so the whole chamber also is made of the plastics. And then that has been connected with the peristaltic pump, where you have to make sure that the blood is flowing, kind of to mimic the blood flow, we are flowing this medium here for the cells to grow. Right, so the whole thing was kept inside the 37°C incubator, as you have seen in the previous video, that the scientist was keeping the cells inside the 37° incubator, which has a blood gas exchange inside. So that has been maintained. So what's the bottom line of this? Here is the 3-dimensional, just to mimic the physiological relevancy, like your endothelial cells, astrocytes, and blood. This all can be done. So that's what the purpose of the three-dimensional. There are many other things have been done beyond this one, but this is some kind of prototype we have made. And so the outcome of the result experiment was that we found that the astrocyte, producing a transforming growth factor, which is called a picomole concentration, which helps maintains the barrier properties in the cell of the blood-brain barrier. So just to recap the cell culture or the in vitro model. So we talked about the two-dimensional cell culture where we have used the curcumin and SARS-CoV-2 model. And we talked about the dimensional just a minute ago, what factors are being secreted from the astrocytes and how it's being, maintaining the barrier properties. Okay, now we move on to the in vivo model. But before we go on to the in vivo models, let's talk about the animal models. What are the animal models that have been used and why are we using, what is the ethics regulation and stuff like that. So what is an animal model? Basically, we don't call it as an animal model. They are the model organisms. They mimic the thing. So, but in a conventional way, in a talkative way, we use the word as animal model. What is that? It's a living, non-human, often rats, mice, guinea pig, etc. And why we using that? Similar to humans. But it means a lot. We can talk a whole lot about that. What the similarity are. I'm sorry. And moreover, it's abundant supply. That's the one thing. And ease of use and high reproductive rate. For example, mice has 20 days of gestation period, and their maintenance is very low cost. And there we have been used to doing research and investigation of human diseases for the purpose of better understanding of the disease process without the added risk of harming an actual human. So the whole thing has been done in the closed controlled environment to avoid the variables. So now next question one would ask, some people would love the animals like the activist which we saw in my first slide. Our lab animals are suffering in pain. Animal use is carefully controlled, particularly if it might cause pain. And there's a lot of laws mandate avoiding alleviating pain and distress for lab animals. Of course, the good science always comes from the animal's health and comfort. And all animal protocols are reviewed and must be approved by committees that include members of the public. In India, we have the Committee for the Purpose of Control and Supervision of Experiments on Animals, which is Government of India organization, which controls everything. Just to give you an example of that in my personal experience, that during my PhD at All India Institute of Medical Sciences, I used the non-human primates monkey model for doing some neurological experiments, and we have submitted the ethics committee clearance for my experiments. And it took almost a year to year and a half just to get clearances. That includes a lot of the scientists, lawyers, attorneys, and publics. So they all will discuss, do we need to do these experiments or not? It was very rigorous, and it took a long time for them to give permission to me, and they changed the whole of my protocol to a different way of doing that. So we have a very good and ethics committee. And not only there, but everywhere throughout the world. So here we have this called the IACUC, it's the Institutional Animal Care and Use Committees, and another one is ALAP. So those are the committees which controls the regulating the how to use the animals. Okay, now just to give a small information that so out of 83% of these Nobel Prize in Physiology and Medicine was awarded to the researchers used animal models in the research. For example, 1901 German scientists developed vaccine for using horses, and very late as the last year, 2021, discoveries for receptors and temperature, were used because of mice. And the next question one would want to know that, are we testing all those the compound which we are taking right now as a drug? Have they been tested in the animal model? Yes, obviously they are all. Like you see some of the few, actually there was hundreds or 200 medicines are there. So here, just to highlight this cholesterol, which we have been checking it, high blood pressure, diabetes 1 and 2. And if you see here, this high cholesterol number 2 has been tested, this compound has been tested in more than 8 animal models before it given to the humans. So that much rigorous we have to do before we get into the human. So now, since I talked about the drugs, so now what pharmacology people are BPharm students, and there's a lot of people sitting there. or watching this lecture, would want to know that how many years would take a normal general question as a person, how many years will it take for a drug to be discovered, developed, and approved a process. In the United States, it will take 12 to 15 years from lab to US patients, or bench to clinics. Hello, can you hear me now? Yep. Okay. I'm sorry. Yeah. As I've been involved in this preclinical testing at the Utah State here, I know something about that. It takes 6.5 years for the compound to be discovered and to be tested on the preclinically. So what we have been doing it for that much amount of time, once it's been discovered in the lab, and then it comes to animal studies, where here we will take almost year and a half or sometimes 2 years just to verify that, animal studies, and we assess safety, what we will be doing that. So we are assessing safety and biological activity and formulations also. Everything has to be done before we move on to the phase 1 clinical trial. So that has been done with animal models. So for example, if we get a compound, we receive a compound, we have to do a lot of assays before it move on to the phase 1. We test for this safety of the animals, and biological activity of the animals, for kinetics, all those things. And then this compound has been moved to phase 1, which is called the IND, meaning that new drug will be submitted to the FDA for the phase 1 clinical trial, which will take almost 1.5 years. And where they will be testing 20 to 100 healthy volunteers to determine safety and dosage of the compound. And then it goes to phase 2, where they will be really using patient volunteers, 100 to 500 patient volunteers. I'll talk about that when I'm doing my next presentation on the animal models. So what happened to one of the compounds we tried. So and then it move on to the phase 3. There they will be testing for 1,000 to 5,000 patient volunteers will be tested, and then it goes on to the FDA for another 1.5 years for review and approval process. What you see here at the bottom of this row, success rate. So when 5,000 compounds are evaluated, out of which 5 only enter into clinical trials and 1 is approved. So that's the time of it takes for 12 to 15 years to approve the trial. So this is a vaccine research which comes the same, but I'm not going to detail, but you would all surprised, oh, then so how did we get this vaccine for the SARS-CoV-2 within a year and a half? But it's entirely different technology have been used for that. We can take a big lecture on that, which is an entirely different topic. But just to give you an insight of that, normally it takes 8 to 14 years for the vaccine to be researched and developed. Okay, now we'll move on to the real in vivo animal models, where I'm going to talk about this. A couple of models where we have used the West Nile virus and Zika virus. Just want to again to tell you that since I've been doing the virus research for this many years, I'll be talking mostly on the virus, but doesn't mean that animal models and then cell culture models only can be used to check for this virus stuff. It can be done, many of the studies has been done to understand basics as well as the disease model. Okay, on the West Nile virus, we have 2 different models. One is the Syphilis black mice and Syrian golden hamster. Okay, so now one would ask, why do you want to use the Syrian golden hamster more often than the Syphilis black? Because we also wanted to study the basics of the virus infections. It's not only when we get the virus, we don't want to see that how to do the treatment. We also want to know how the disease is progressing. What's the pathogens of the virus? What is the second level? What's the next level? What if a person has infected? What will happen in many years from now? Something like that. So to do that, we chose to use the golden hamster, which showed the which exhibit the disease signs close to the humans. So that's why we chose that. And the next one, the Zika virus, where we have used the AG129 mouse. You want to know what's A and G. A is alpha and G is a gamma. Interferon knockout mice. And if you know some of the immunology, you would know that, oh man, you already removed the innate immune system, the first level of defense, you disabled that. And why do you want to use that? Immunodeficient mice, it's already been compromised. It's already been, prone to be infection. So why do you want to use that? Because the Zika virus model cannot be developed in the wild type animals. Say, for example, the wild animals will not elicit any disease. So we have to have some kind of model to start the treatment. So that's why we use the immunodeficient model. Many of the viruses have something like this. So we have to use the deficient model, or you have to treat it chemically to become deficient, and then you start your treatment, or you start with infection to elicit. So that's the purpose of using this immunodeficient model here. And now let's go to the West Nile virus. What is that? Which is started in 1937 from West Nile virus to Uganda, which is the single-strand virus. What is similar type of virus? The Japanese encephalitis, dengue, and Zika are the similar type of viruses. And it has been transmitted with the culex. We have a culex everywhere in India. Everywhere, but specifically, I want to kind of let everybody know that we have a lot of culex. I work with the culex mosquitoes for my master's degree project. So they are very notorious. They go everywhere. So what kind of disease we'll get with West Nile? Fever, headache, vomiting, diarrhea, muscle aches, and rash. And the complications would be the encephalitis and meningitis, leads to respiratory complications. The usual onset is 2 to 14 days after the exposure, and duration, it goes from weeks to months. The diagnostic method based on symptoms and blood tests. And very sadly that there is no prevention or there is no treatment for that. Now, what it has been suggested in the CDC website, Center for Disease Control here in United States, they say that wear long sleeves or and use insect repellents. So that's the only prevention we have. There's no other medication for that. There's no antivirals and there is no anti-vaccines for that. That's why we have been working on that to find out something. So the prognosis is 10% risk of death among those seriously affected. That's one of the very serious conditions. Okay, so let's see what is the normal transmission cycle of this virus. It's mainly in the circuits between the mosquito and then these birds. Occasionally it infects the humans and the horse. They are the dead-end horse. Excuse me. And what situation in India? Based on this publication on 2021, very recent, they found that West Nile virus has been found all over India, Madhya Pradesh, Kerala, West Bengal, Uttar Pradesh, Assam. And in Tamil Nadu, Madurai and Sadiperi areas, I found more cases, and Karnataka, Maharashtra, and Chhattisgarh. This is 2021. And you also might have heard that in Kerala, very recently, this one was published in the May 31, 2022, a couple of months before. The Kerala department is set on high alert to inform the people that be careful with the virus. It's there everywhere. The mosquitoes are everywhere, so you have to be careful. So, any virus disease can be compared with analogous to the crash of the Titanic ship. When the captain saw initially a small iceberg, it's kind of initial infection that we call acute. But he did not know that there's a big iceberg below that. Like you see here, a big iceberg, which is kind of analogous to this one. So initially we saw that, an infected patient will have this fever, headache, chills, and weakness. But whereas the long-term infections, they will have a lot of neurological complications, neurodegenerative diseases, and the respiratory complications. So here, when we infect these Syrian hamsters, so they will display or exhibit a different kind of disease, like a diarrhea, tremors, eye lacrimation, and combination of paralysis, like some, which is very similar to human disease, human will exhibit paralysis. And they also have the neurodegenerative complexes also because of these infections. But 10% will die without no signs. And very interestingly, just to think about everybody, 20% will alive without any signs. It's like something like a human infections, like when we have the SARS. Not everyone got the SARS. Very few got the SARS, and many people survived. So something innate immune system is very even in the animal. But one would also ask that, oh man, your animal model is not lethal model, so that's why you have the 20% survival. But for everybody's information, we tried different stocks of the virus, different dosage of the viruses. We get to see something like this, particularly with this model. Because we have another model which is 100% lethal, whatever the virus, even if you get one virus particle, the animal will die. So maybe the Western virus is not that lethal, possibly. So what is the time course of the infections of the virus? So here, when you infect on day 0, the virus will become peak in the serum. So that's called the viremic. Serum virus number will be increasing. The viral titer will increase. That's called the viremic phase. And then once the virus declined on the serum, then simultaneously you will have some of the virus particles or the virus infected cells or virus infected cytokines will cross the blood-brain barrier, as I showed in my in vitro cell culture model, 3-dimensional model, the blood-brain barrier. Remember that? So that's where it crosses the brain, will infect the brain. Okay, so there it's called acute infections. You can have a complete recovery from these both conditions, viremic and acute CNS infections, and some will die, unknown cause. And the people, those who have the virus for a long time, will develop the neurological sequelae. So that's called the persistent Western virus infections that can be correlated with the lesions, with the cause and effect. Okay, so what we have done here as an antiviral group, so we use this antibody called HE16. This has been prepared by one of the collaborator, and it's also been done by a big company. Thought that they would make it as a human trial, as like the same step as they've gone into that. But because of that, as I showed in phase 2 clinical trial, they have to have a minimum number of patients to test this. Since this is the seasonal infections, they didn't get enough number of patients, so it cannot go on to the phase 2 or phase 3. Didn't pass the phase 2. But we have done a lot of work on this, hoping that this will become some of the antibody treatment for the Western virus infections, but which did not. But either way, but what we did, okay, here what you see here, this compound, this antibody has been treated on the 5 days after the infections. Now, so this is a D and E of the same graph. What is the A, B, and C? Okay, A, B, and C are the different time of that. This is day 1, day 2, day 3 treated. Those are all 100% animals survive. So now you have a question. So why do you want to treat on day 5? When a compound has been tested positive or the protective effect on this prophylactic treatment before the infections, more animal models, not the real world. So we want to see how long we can delay the treatment, because most of the time patients will come to the clinic after the infections, and moreover, once you get to the clinic, you have to diagnose what kind of infections you have. So that all will take time. So once we find the compound which is working, so we want to know how long we can delay the treatment. So that's a very big question all the time. So we delayed the treatment of day 5 and day 6. As you see here, day 5, which is clearly protected the animal. 95% animals have been survived when compared with the placebo-treated animal or saline-only treated animal here in the line. But 6 days post-infections, which did not help, even though we administered 3 times more than this one, 100 milligram per kilogram once. So this paper also been published here at the bottom. What you see, all these things published are the manuscript which I got these references. Okay, like I said earlier, West Nile virus infect causes the diseases. That means it's also infecting some part of the brain. So we want to know how can we treat the neurological diseases just by providing the antibody directly onto the brain. So we have used a technique called convection animal delivery into the brain. So let's see that what does that mean. A small video for a minute. Okay.
Alzed osmotic pumps have no batteries, no electronics, and no moving parts that can fail. The pumps are self-powered through the reliable process of osmotic displacement. Here's how it works. Alzed osmotic pumps consist of 3 concentric layers: the inner drug reservoir, the osmotic layer, and the outer rate-controlling semi-permeable membrane. The reservoir within the core of the pump is filled with the test solution to be delivered. When the pump is implanted, water is absorbed through the outer membrane. The water entering the pump expands the osmotic layer, compressing the flexible impermeable reservoir. This enables the test solution to be released through the exit port at a controlled, predetermined rate.
Yeah, so that's how we delivered that antibody to the brain. And again, we delay the treatment of 5 days post-infections and 6 and 8 DPI. Excuse me. Sorry, there's some noise coming from outside, so I just want to close the door. Okay, we treated them on day 5 and day 6 and day 8 on the different areas of the brain. And when we treat that at day 5, which is something like intraperitoneal treatment, it's again prevented the animal from the death at 5 days post-infection. So we concluded that, so either you administer by the intraperitoneally, or if you administer by the convection means delivered directly to the brain, which protects the animal at the 5 days after the infections. Okay, now we move on to the next virus, Zika virus, which is from Uganda, 1947, which has the same similarity like dengue and Japanese encephalitis viruses. Again, which has been caused by this different type of strain of mosquitoes like Aedes group, Aedes aegypti, Aedes albopictus. And the very notorious thing was it can also transmit vertically transmission. What is a vertical transmission? Pregnant women, if they get infected during the, I'm not sure about the first trimester or second or third trimester, but at one time point, if they get infected, the virus can pass to the fetus. That's very dangerous one. And it also can be transmitted sexually. It also has been transmitted blood transfusions. Probably you might have all heard that since 2014 and 2015, we had a big outbreak of the Zika virus all over the South America here, and it also came to the Americas, and we have the cases in India to talk about as we move on to the next slide. Okay, so the symptoms is fever, headaches, vomiting, diarrhea, and it also can cause encephalitis. Microcephaly is the smallest size of the head. Micro means small, cephaly means head, a smaller head. And Guillain-Barré syndrome is another kind of immunological disease which has been caused by this virus. These will answer 2 to 14 days. And then based on symptoms and blood tests only we can diagnose this. And again, the prevention and the treatment, nothing is available. So only thing that the long sleeves use insect repellent. But one very interesting thing is that I think there's some company in South America, they genetically modified the male mosquitoes. Okay. When they made the females, after the female laid eggs, and whatever the hatching eggs will become only the male and not the females. The question is that what's the female is doing? Because a female mosquito needs a blood meal to produce an egg. So that's the purpose of that. So the female only bites the humans, whereas the male mosquitoes take food from that tree sap and like that. So that's why they wanted to make this one, and it's successfully done. And they also, I read on newspaper that they released in wild some number of genetically modified male mosquitoes, and which has really helped them. But unfortunately, we don't have any vaccines or antivirals. We only have supportive care. Death is very rare. Now look at the cycle. So mainly it's found in the non-human primates and the mosquitoes in the wild, but occasionally it infects the humans, and then humans can transmitted by the blood transfusion and sexual transmission, and it can come to the perinatal transmission like congenital through the infected mothers, and they can pass on to the babies. So what's happening in India? Very good question, right? So India has the largest Zika exposed population in Asia, and the country has almost 289 cases 2018, up from 2016, 2017. So these are the red colored areas where we have found the virus, I'm sorry. And in 1954, the first case was reported in the Gujarat. And then after that, it started coming back in 2016, 2017, 2018. And we also found a person with this positive in Tamil Nadu also. And then it's become outbreak in 2018 in Madhya Pradesh, where they had a lot of 930 cases. So Zika passage to India. So after this many circles, like 2015, Brazil reports an unusual increase in microcephaly cases. Here I showed you that a small size of head. I will talk about that later. So, and then it came to India 2017, and in 2021, last year, we had the more cases. But what is the, when do we see this virus most? See the virus most when we have the more mosquitoes, right? So what is the mosquito population time when they will become eruptive, when they will become seen more? So we'll see more on during this May, June, and July. So that's where we see the more cases in Rajasthan and Madhya Pradesh. The case is very high. So we have to be aware of this also, Zika virus. Okay, so what we have done as an antiviral group. So we have used the Malaysian strain of Zika virus. There are so many different strains of viruses, but we have used a Malaysian strain. And again, we start with the animal model, as it showed you that we have used a type 1, type 2 interferon receptor mouse. I already gave you the why we have used that one, why we used that. So, and when we infect that animal, we see that this is Zika infected animal, all the animals will die by day 24. And this is sham infected animal. And they also show a lot of disease signs like excitability, lying prone, paralysis, hunching, and conjunctivitis, all those disease signs. So what we have done as an antivirals again, so we use the BCX4430, which is a compound nucleoside analogs. And probably pharmacy students will understand what does that mean. So this compound is originally is a galidesivir that has been approved for the human hepatitis C virus. This as a repurposed drug for these emerging viruses like the Zika virus and yellow fever, Ebola, or Marburg. So this has been already commercialized and is repurposed for these different kind of viruses here. So here we are looking at this when we treat the different doses of the BCX4430, 300 mg and 100 mg per kilogram. We saw good efficacy, protective effect when we use 300 milligram. So now the next question, as I told you already, how long we can delay the treatment. So we did the same thing here. We found that this, we can delay up to 1 day after the infections, and then after 3, 5, 7 days post-infection, it won't survive. They will die. All the animals have died. So here's another very sad picture to show that. So these are the twins born with the Zika virus infected mother, and they have a case of microcephaly. So that's the sexual head you see here. On your right side here, you see that the live-born infants in the United States with the Zika virus repository, where they found 6,700 kids were born with the virus infections, on which 252 kids had the microcephaly, and on which head circumference were reduced 3% in 27 cases. So we tried to mimic the same thing in our animal model using the AG129 mice, and when they become pregnant, after 7 days of the pregnancy, we infected with the virus, and after the babies were born, they were fixed, and we did the micro-CT to see was there anything correlation of that microcephaly to the same thing here. So the skull length, we measured the 4 different parameters: crown rump, skull length, cranial height, and biparatal. Like you see here, all these parameters. So the skull length was reduced, like this one was reduced, shrunken, at a statistically significant level. You see in millimeter size, but is that a millimeter really makes sense? Yeah, even a 1 micron difference in the size of the head will make a lot of difference. So how this is affected? It's basically this Zika virus system delaying the cell divisions during the time of the pregnancy in cell, and that leads to the further proliferation of the cells. I mean, decreasing the further proliferation of the cells. So that's one of the reason here, elongation of the cell division by the Zika virus, leading or causing the microcephaly cases. So just to recap my presentation so far, I think we come to the end. So we talked about the in vitro model where we use the 2-dimensional cell culture, see how the curcumin is preventing the SARS-CoV-2 using that model, and 3-dimensional we use that was a diseased model, and 3-dimensional cell culture was the basic science. We want to know that how we can what's the role of the astrocytes in maintaining the barrier properties. And in the in vivo model, we have done this, we studied the wild type hamster that's infected with this virus, West Nile, and then what we have treated. The next one was the Zika virus, where we have immunocompromised animal, a different one. So I think I just covered the how we can use the both cell and in vitro and in vivo models for studying the basic and diseased models. So I think I'm completing my presentations and make an entry, and I'm going to take any questions. Hello?
Yeah, now the session is open for discussion. This is Q&A session.
For the Q&A session, I already put the feedback form onto the chat box. Okay, Namanita. Questions? Or you can also put your question in the chat box if I cannot hear you or you cannot talk to me. You can see Namanita can raise the hand, but I don't see any questions from them. I cannot hear you. Or you can type your question in the chat box. Yeah, I see you're raising your hand, but
Yeah, you have to mute him, I think. You have to mute him.
okay, so how can I do all in one?
Excuse me, Mr. Navanidham, sir. Can you put your question on the chat box?
Hello, sir. Hello.
Yes, yes.
No, just no. The curcumin you are telling, sir, beta diketone isolated. More other than that, any other compound has been isolated, sir.
Say it again. I didn't get you. What was that question?
No, curcumin. You are telling, that beta diketone. That this side is phenyl group, 2 dihydroxy groups. That one compound you have shown, sir. The structure,
Yeah, structure. Yeah. Curcumin. Yeah.
Yeah. So actually, whether it is available, only the main compound is that one, or other than that, any other compound has been isolated, sir.
I think I'm not experienced on that. I can ask Mr. Shanmugam to answer the question. Polyhedron.
Oh yeah. I think he has done a lot of work on that, I think.
Yeah, I mean, he would be appropriate person to ask the questions because I'm an end user. Most other compounds. So
no, just I want to know, sir, whether it is the maximum isolated compound is that one or only minimum. What is the percentage of the compound available in the curcumin, sir? That just I want to know.
Okay, I think Dr. Shanmugam, can you answer that?
Sir, you have to unmute everyone.
Yeah, that's what I'm trying to say.
Yeah, you have to unmute everyone. Then only sir can ask. Sir can answer.
Yeah, so there is that unmute box here. These are long presentation, so I've done this one.
Hello. Can you hear me now? I am Siva Shanmugam.
I can hear
Yes,
you.
sir.
Hello.
Yes,
Here,
yes,
sir.
sir.
See, so from turmeric, when we extract, we normally get curcumin,
Mhm.
and curcumin is having two methoxy groups.
Yeah,
So,
that,
along with the curcumin, we will get one molecule which is having OH group and another is O-methyl group. That is called dimethoxy curcumin. And the third one is both methoxy group will be removed and they will be replaced with the OH groups.
Okay.
So generally speaking, curcumin will be 75% in that mixer, and dimethoxy curcumin will be approximately say about 15%, and bis dimethoxy curcumin will be approximately 2 to 5%. And
Okay.
when we extract out of the turmeric, when we crystallize it, we'll be having the mixture of all the three.
So how you are separating, sir? Are these three compounds?
Actually, it is a solvent, pure solvent extraction. Normally, the solvents earlier it was used. Earlier solvents were dichloromethane or dichloroethane. Now it's banned. Chlorinated solvents are banned. So we are using ethyl acetate for extracting it. After extracting it, we are recrystallizing it using ethanol or acetone.
Oh, it's just by fractional crystallization. Fractional crystallization, you are separating.
We cannot say it is a fractional crystallization. They all crystallize together. We get as a co-crystallization only. We get always three components together. These are all alkaloids. And also we are having some aroma in it. That aroma is mainly due to the compound, a terpene called turmerone. So turmerone we are normally taking away by something called a steam distillation. So we get both turmerone as a terpene and curcumin, dimethoxy curcumin, bis dimethoxy curcumin as a co-crystals all together.
No, just sir, I want to tell you, sir, one thing. Just to hear the dihydroxy, monohydroxy, that can be separated by using dilute sodium hydroxide, sir, at cold condition. So when compared to dimethoxy, only dihydroxy or monohydroxy will be soluble. I think by way.
Can you please repeat once again?
No, that.
Can you please repeat once again?
No, sorry. If the mixture is soluble in sodium hydroxide, dilute sodium hydroxide.
Yes, I have seen it.
All hydroxy compounds will be soluble in sodium hydroxide. If you use some solvent, that dimethoxy compound can be extracted. That can be easily separated from the hydroxy compound.
Excellent. Very good.
Yeah, by that method, I think we can use that method, to separate the three isomers.
We'll try.
Yeah.
We will try tomorrow.
Okay, sir. Thank you,
sir. Excellent information. Thank you, sir. Thanks a lot.
Okay, sir. Thank you.
Actually, your voice is seen, but nobody has seen you. This is Dr. Rajendra Prasad, former HOD of Chemistry of Bhajan University.
Oh,
okay. Dr. Siddharthan, this is
sir told me, sir. Dr. Siva sir told me, sir, about you, sir, your achievement and all.
Okay, thank you.
Sir, thank you, sir, very much. Your presentation is very nice, sir. Very excellent.
Thank you.
It is actually, we came to know several things, sir, about this virus infection and all, sir.
Okay. Thank you.
Thank you very much.
Any other questions, please?
Yes, this is Navanidam again. I could unmute the mic. So, Dr. Siddharthan, do you hear me now?
I can hear you. Go ahead.
Hello?
I can hear you.
Do you hear me? Okay, very nice talk. You know, regarding that Zika virus, a very little question. These kids who are born with Zika infection or Zika infected parents, they have microcephaly, right? They physically look fine, but what is the consequences as they grow? What happens to them, those kids? Intellectual problem or physical problem?
Yeah, it is a big question. Actually, we all have been wondering about that. It's not too many years we passed the Zika phase. It started in 2016, 2017, and many of the data we are still getting. in from foreign countries, but I, here and there I can hear that this is, we don't get the full detail of the data as of now. So we may come to know maybe in a coming years, maybe a few years from now, we'll know that what's the fate of those kids. And we cannot able to try to develop any animal model for that because we have done so many different types of infections because in the animals, particularly the AG129, as I told you earlier, it was really difficult for them. We infect directly the pups. We cut open the placenta and we try to infect the pups and put it back, and then sometimes it's kind of they all kind of won't deliver or they will be. We don't have any evidence of that, but there is something which I have used that we did that. The pups which were born on the infected dams, we let them grow for many months, and we have done this auditory testing. That's called the brain auditory evoke response. That we have published a paper on that. So I have done this. It's kind of in-house development of checking the hearing properties. Can be able to hear with the high and low decibels. So not all the pups who are born to that with the virus, and very few had this hearing deficiency. That much I know basic, but they were looking some level. They were looking physically normal because I showed you that there are some millimeters difference in the skull length. But they had some hearing deficiency from our experience. I'm guessing that there will be a lot of things for the kids and still it's going on for the children who born with Zika.
Thanks. Good to know that. So being a underdeveloped brain, smaller cranial cavity means a smaller brain. So one can expect as they grow, there are a lot of physical disabilities for them.
Sure.
Thanks. Very nice talk. A lot of information. Thank you.
Thanks, Samita.
Thank you so much, sir. You have one more question in the chat box.
Okay.
You have one more question in the chat box.
Yes.
Can you able to see, or I have to read it out?
I can read that. Is it from Shakti or?
Yeah. Yes, sir. Yes, sir.
Is it possible that the question is it possible that vaccines are good, effective, which you discovered and approved within 1 to 2 years? That's a very interesting question. We all have been wondering about that. That's why we are having more, what's it called? Booster shot every now and then. Every 6 months we have to have it. So, so far it is good. I won't say it's not good. So far it is good. So far it's working very well. And but how long it will be effective? So that's a big if. And that's why we have to have booster shot. They are good. I won't say no good. They are good.
Can I ask one question related to that?
Go ahead.
Yeah. See, we are very much concerned about the safety of the vaccines what we had, and we came across so many things, so many experiences that they developed some sort of side effect, something like rheumatism and all. Is it really true, or it is only a myth? The one what we had for the this COVID.
What is that? What was that last word?
See, the vaccines what we had for the COVID, many people are reporting that they developed some side effects like rheumatism and all.
I got it.
Is it true, or
okay?
Is it a right thing, or it's a myth, or what is it? What's your opinion?
There's no myth on that because if the patient have a problem, so that means true. So, but I would say that it's a very rare event. I mean, I have had so many cases here in even within my friend circle, and even then the CDC, they have a different website that, if one have the vaccine and if you have a problem, you can go on, log in, or you can put your information there so that everybody can know that. So they have some kind of feedback system at the CDC. So I've heard that people have the different kind of problems. It's not only the rheumatism. They have some will have some kind of fast heartbeat times, or some other complications. There's no between,
So you mean
yeah, go ahead.
to say that it is not really met? There could have been a problem. Am I right?
Right.
Fine. Thank you.
Sir, you have one more
Hello.
question from Mr. Chandru. Chandru, I'm unable to unmute yourself. Can you put it in the chat box?
Chandru.
Mr. Chandru,
Hello.
can you put it? Can you
Hello.
put your question in the chat box?
And there is some, yes, Ravi had a question on the chat box. Is it possible to have
Yeah.
conversation with you to test our synthetic compounds in your end? Yes, of course, definitely it's possible, but we have a system called the, everything has to come through the NIR, which is called the National Institute of Allergy and Infectious Diseases. That's a part of the National Institute of Health. So it has to come through the proper channel, and then we can able to test your compound. So yeah, I mean, maybe you can communicate me directly by email. I think I had my email on the first slide. That,
Sir, you have 2 more questions in the chat box. You
okay.
have 2 more questions in the chat box.
Okay, Manil, I will read the question. Now I'm answering this question to Mr. Ravi. Yes, you can email me separately and we can talk about that, Mr. Ravi. Thank you. Okay, so what's other question?
Yes, Mr. Ravi, I can share his email ID to you. You can have from me and you can communicate with him.
Yeah, maybe like
Another question I have, it is in the chat box. I just like to know there is one question. What is the reason for the second attack when there is a natural immunity is developed after the first attack? There are many people who have got the second attack. That is one thing. Second, even after having vaccination, many people had it. Is there any reason for it or any valid reasons behind it?
It varies from individual to individual. That's what I see. Because based on the animals, we haven't done any animal studies for a long enough to see recurrence of these, virus, like a tumor, but that's not possible with the virus infections because you have to have a secondary infections, you have to have new infections to have that. And moreover, nowadays we have so many variants of the virus. Like we have a BA 2.5, which is in India right now, and BA 2 and BA 1.75, like the Delta and Omicron. So I don't have any clear-cut answer for that right now, why people getting secondary infections or again infections. So yeah.
Thank you.
Not answering your question straight away, but I'm kind of like everyone.
Thank you.
Since we are all learning the new things. Okay, there's a question from Jay Vignesh that can we do the how they did the co-infection, post-infection, how the virus moved the cell before introduction of curcumin. So first of all, these are the 3 different experiments: pre, pro, and post-infections. All different set of experiments. And then how is the virus removed from the cell lines before introduction to curcumin? Yeah, you use the same kind of pipetting techniques, and then you take it out. And then you dispose the virus carefully because these are the SARS-CoV-2. We have to dispose them in a separate way. Yes, it's possible. You remove the virus using the pipette. So it's all done inside the BSL-3 level laboratories, and you will have all kind of respiratory masks, and you will cover you have a separate respiration system for yourself. So yes, it's possible. Okay, so what other questions?
Hello.
Yeah, Jay Vignesh. Who else has a question now?
I am Ravi from Karpagam Academy of Higher Education.
Okay, so what's your question, sir?
Yes, sir. That is usually in India, it is very difficult to work the viral studies.
Yeah.
And we are the synthetic chemists and we do a lot of compounds. And for a budding scientist, to what extent he can do the experiments? What advice you would like to give?
Okay, I think in India we have this National Institute of Virology in Pune, and we also have one more institute in Delhi. I think in Old Delhi area. I'm not sure. what's the name of that? So there, these kind of compounds has been tested. And I also see some publications from some from Kerala lately. I'm not sure it's the Chittarethinol Institute of Medical Sciences or somewhere they have been doing some kind of, particularly with the curcumin studies, they've done a lot of these turmeric studies. And specifically with the virology using synthetic compounds, very few manuscripts from India. As I told you, if you wanted to test your compound here in our system, it has to come through a proper with NIH and NIAID. So I gave my email ID on the chat box, so you can send me the email, and then we can I can maybe connect with you somebody in the NIH or NIAID so that your compound can be tested.
Thank you, sir. Sir, once again, even though we have 2 national institutes which work on virus, normally there will be a heavy rush and they will prepare the compounds from IASC and similar institutes like that. So suppose if you want to collaborate with you, before sending the compounds in your end, what type of studies we should carry out and we should ensure so that it will work on these studies and confidently we can send it to you.
Yeah. Can you give some times for me to analyze whatever the question you have asked so that I can answer to you accordingly, so that we can have some communications established.
Yeah, thank you. Thank you, please.
Thank you, Ravi. Thanks. Okay, anybody else have a question here?
Sir,
Go ahead.
curcumin has a safety lagging in research. Confirming studies have been done by the World Health Organizations. How long you recommended the doses for the curcumins will be happening in the during phases?
During the?
Actually, the studies was saying that safety is lagging in curcumin, while the recommendation of long-term dosages.
Yeah, I mean, again, I am kind of a little lack of knowledge on that, what kind of pharmacokinetic studies have been done on that. I think if anybody there who has tested this compound for the pharmacological studies can give some insights to Mr. Chandru.
I can share some details to him if he shares his email ID. I'll take his email ID from the registration form,
Okay.
and I can share some details to him. Right now, I don't have, being a chemist, I don't have right answer for him, but have relevant papers with me. I can share with him.
Okay, for everybody.
Is it audible?
I put the feedback form chat box. Can you guys see that? It's at the #1 chat, first chat at 6:54 a.m. PM. There's a form there. Feedback forms. The Achara Pandian Zika virus able to cause hepatitis. We have seen the West Nile virus infecting the intestine, but we haven't seen anything like a infection of the lung or the liver. Yeah, liver. So I'm not sure, Mr. Acharapandian. I'm not sure about that. We haven't done anything.
Hello.
Is for this kind of a normal, it's kind of our painkiller, acetaminophen. So it's another painkiller. That's it. So it's not any. I'm not sure it has antiviral or any properties. Chandru, I answered your question. Chandru.
Not Chandru.
Hello.
Hello.
Sir, one more question.
Go ahead.
While you work with curcumin, are you satisfied with the bioavailability of curcumin?
Again, this question goes to Shiva Shanmugam. Can you answer that question, sir?
No, you may know that is curcumin works well, but the bioavailability, it should be done if that is during the in vivo studies.
Yeah. Basically, as an antiviral group, we don't do that kind of studies because that's not supported in our program. But I can find out maybe and let you know that. Is that Dr. Rajendra Prasad?
No, Dr. Ravi.
Right, okay. We'll find out and let you know.
No, I will
Yeah,
give his
thank-
email ID to you.
Yeah, okay.
I noted on his question. I will share it with you.
Okay.
Thank you, sir. Thank you. Thank you.
Any other questions from any participants? Any other question from any other participants?
Yeah, I have questions too. How many of you people like this presentations? Was it informative to you all? Or was that?
There is one question from Arul Raj: how to reduce the toxicity in the drug compounds? Is it relevant to your area of research?
Likely not. Any toxic can you help him, Arul Raj, to know that toxicity of the compound?
W magazine money.
One second, please.
Arunika Nagarajan, can you mute yourself? Arunika Nagarajan, please mute yourself. Yes,
There
sir.
is one more weird question from Jay Vignesh. Why is that most diseases originate mostly from Africa? Any known answer for that?
I don't know, but one can assume that that's the I'm not a philanthropist or something like that, or philosophist to give some kind of question for that. It's not the, it's like you said, it's a weird question. We had this COVID-19 from China recently.
Okay.
We had these, other weird viruses from these
Singapore, we have SARS.
Yeah, we do have a
Yeah.
SARS. Now we have
Middle East, we have
MERS.
MERS.
That's
Yes.
another SARS family, MERS. Syndrome.
Right.
Zika was.
Is there any logic? Is there any logical answer for where it develops, why it develops like that? Anything like that? Because till last 2019, we never knew anything about Corona.
Yeah.
But suddenly there was an outburst. How they originated from, what is the genesis of that virus? Is that, although it is out of the topic.
Kartika Ramamurthy. Can you mute yourself? Yeah, thank you.
Yeah, my question is over. Is there any, can you define, or is there any what to say, the logic, how they are originating newly, suddenly? Is it a mutation of some other virus, or it is something newly getting generated?
I think they're all in the animals for many, many years. And once it's get into the human, for example, if you take the SARS-CoV-2, so they say that it was in the many of the different animals. We do not know was it a pangolin, was it the bat, or was it something else? They all residing in some form, in a dormant form, or in the non-expressive mode in the animals. Okay, but when they get to the human, the first pass, the first person contact with the virus, he won't have any infections, because what the virus is doing is trying to mutate, trying to adapt its life on the human. And then it's just move on. When the next human was infected with the virus, he will be having more serious disease than the first person who. So the virus is mutating inside.
Moganapriya, please mute yourself. Moganapriya, please mute yourself. Thank you.
I think that's how it's been. Okay, there was one question from Teshini to Okay, any particular reason for plaque assay being used virus quantification? I mean, that's a golden standard assay which has been used for many, many years. And most of the virologists would want to do the plaque assay only to do the quantification of the virus. So this is not a particular reason. That's the only thing which worked. That's the only thing which is a gold standard for these virologists. So that's why it has been used to quantify the virus.
Good. Thank you. Dr.
Any other
Monvili,
questions?
is there any other questions? Yeah. Can we
Wind up.
Yes. Can we take up? Can you please?
Dear participants,
thank you very
the
much.
feedback link is available in the chat box. You can fill up the feedback form so that you can receive a e-certificate. Feedback form is available. Kindly fill up the form and so that you can receive your certificate. So if there are no questions, one more question in the chat box. Feedback link is okay. Okay. On behalf of GPI, it's my duty to deliver a vote of thanks. Thank you, Dr. Venkatraman Siddharthan, sir, for readily accepting our invitation and coming up with this first wonderful session, an excellent talk on basic and disease biology, usage of models. Thank you so much, sir. My sincere thanks to all the luminaries of the GPI organization for their constant support, mentoring, and endless enthusiasm. I wish to thank all participants from different trust area for their gracious presence here today. Knowledge has great pioneer and has no barrier. Learning does not finish with classroom. It is better to learn young friends. Wishing you good luck. Thank you so much all for joining the first GPI Bridgewell webinar meet. Thank you so much. Thank you so much.